Everybody, welcome to the May Exceed ECSS Symposium. So our title today is COVID-19 Drug Repurposing Guidance Using Fragment Molecular Orbital FMO Calculations. And our speakers are Aaron Frank, who's an assistant professor at the University of Michigan, and Dimuthu, Wannipurage, and Suresh, Marru, who are both with the Indiana University Pervasive Technology Institute. Just a reminder that this is an Exceed event, so we were governed by the Exceed Code of Conduct. I'll advance my slide here. I don't expect everybody to read the entire thing here, but if you have any questions, go back to www.exceed.org/code of conduct. And then I guess we don't need any more. Sorry, that was an earlier announcement. So with that, it looks like our participant list is leveling off. I'm going to stop sharing and I will turn things over to our speakers. They've said that they are happy to take questions as we go along. And I guess since there are three of you, you could answer a lot of the questions through the chat as we go. And then I'll also look for opportunities to interrupt if necessary. So again, let me hand things off to speakers. And I think, Suresh, you're kicking things off.
Yep. Thanks, Bob. And thank you also for giving us the entire hour here. We have an interesting topic and different perspectives and multiple aspects of the project we would like to share. So having this extra time helps. So here we are excited to share a project by Professor Aaron Frank and his collaborator Indrajit Deb who is now at Michigan State University. An interesting project which came through the COVID consortium. If you're not familiar with it, like the US government, the Chief Technology Officer's Office and Office of Science and Technology and Policy quickly put together a consortium of which Exceed participated in, and so did some of the site providers here at Indiana University, for again, the University of Texas at Austin, the TAC group, the couple of resources we used for this project. So when Aaron requested a scientific collaboration service through the ECSS program, so with the normal life cycle, the way it works, like ROP-Quick, it came down to ROP-Quick because it has gateway aspects and it required a multidisciplinary team to help out Aaron and Indrajit execute this project. So given the timely nature of this, as we all know, COVID was rapidly coming on. And like, for example, I'm personally dealing with the families and extended families in India still going through that. And we knew the relevance of it. And so that's one of the reasons, we would like to share both aspects of that today, with Aaron presenting the scientific aspects and how the fundamental research they do can potentially has a future impact in finding drug targets for COVID-19. And we'll also have Dimuthu share how we help from the Exceed side, from the ECSS aspects on orchestrating a lot of computations. So just before I hand off to Aaron, I would like to take a minute or two to set some context here. Here we'll have a long talk with the multiple multidisciplinary aspects. And we see this quite often, in Exceed ECSS program, and when projects like this come on, we have to take help from the resource provider in optimizing codes, like the flagship of what the ECSS program does. So in this case, John Kazis and Kent Melfield help from the Texas Advanced Computing help optimize the code in this the game's quantum chemistry code. But there's a lot of other things happen as well. For instance, Sudha Pamadigantam, who understands the science, which Aaron and his team are doing. It takes some close collaboration, understanding the scientific goal, and so that we can make some context on what are the computations they are doing, what the post-processing, what the life cycle of the science essentially, what kind of computations they will run, what will happen after the computation, so that we can take those post-processing in mind early on and accordingly run through the computation. And this project has certainly required even further aspects, like we had to do some initial testing on the Jetstream cloud at IU, and Eric Coulter, who specializes in quickly putting together virtual clusters on Jetstream, how we can create a cluster environment, try some various versions of the codes, and then hand it off to the big workhorse here, the Stampede 2. and Aroma basically like who manages the gateway, and how she pulled together a gateway quickly so that Aaron and his team can download the results and without monitor all the day-to-day executions and so on. So this is a project where it's a classic representation of what we call the full stack cyber infrastructure, going all the way from the science aspects to the infrastructure and everything in between. So we'll share all of those findings today. And so feel free to ask us questions on the chat. There are those who are not speaking, we'll try to answer them. And then if needed, we'll interrupt the speaker. But the way we'll spend the hour is like, first Aaron will speak the scientific aspects, and then we'll take a big pause and take questions. If you have on the goals and how it's relevant to the COVID and the future work on that. And then Dimuthu is going to take it from there and talk on the distributed system, science gateway, middleware aspects. With that, let me turn it to Aaron and get started here. You're muted, Aaron. Yes.
Okay, so let me share my screen. Can you see?
Yes, we can.
Yeah. Okay, so as Suresh kind of introduced, right? This project is centered around some of the work that we did as part of the COVID-19 consortium, HPC consortium. And so the project is centered around doing drug repurposing, and we use the Fragment molecular orbital method. And I'll kind of give you some more details about this method in a bit. So the work from my end, on my side, was mostly executed by engineer Deb, who was a postdoc in my group at the time, and we're at the University of Michigan. Just a quick background about me. So I am an assistant professor in the biophysics program at the University of Michigan. My expertise is in computational biophysics, and so the work that we do is centered around doing a lot of calculations and simulations of biomolecules. And so here we kind of leverage some of our interest and expertise to try to guide this drug repurposing effort for COVID-19. So I'll start with an introduction. And so I don't think I need to go over these numbers really, but I think it's important to kind of give a scale of the magnitude of problem we're faced with, right? So we're all aware of COVID-19 being a devastating disease. And so here what I'm just highlighting is some of the numbers associated with the pandemic, right? So we have close to 162 million people have been infected by or are affected by COVID-19. Of those, we had 141 million people recovering, and then we had close to 3.3 million people dying from this worldwide, right? So these are really staggering numbers, and they kind of really capture the magnitude of this pandemic. Just in the United States, right, the number of confirmed cases is about 33 million, right, with 27 million people having recovered, and then about close to 600,000 people have died, right? And so despite all the resources we have in the US, right, we still see that COVID-19 has really had a significant impact on the public health, and not to mention its impact on our daily lives. So naturally, there has been a lot of effort centered around trying to come up with therapeutic approaches to deal with COVID-19. And you're all aware of the vaccines, and I'm pretty sure many of you have received the vaccines. But we still know that there is a case of vaccine hesitancy. And so there is also a need to develop or identify drug molecules, conventional drug molecules that can also be used to treat people who might have COVID-19. So here I'm just showing you the life cycle of the virus, right? This is a pretty busy slide, and I'm not a biologist, so I'm not going to go through all the details, but it's just meant to kind of give you a sense of the general strategy, right? So we have the virus outside of the host, right? And it binds to the surface of the host cell, and it goes through a series of steps, and eventually it's released. And so the thing that I wanted to highlight are these pink boxes that highlight some key points where you can actually intervene, right? So there are these host proteases that one could imagine identifying inhibitors for, right? And there are also viral proteases that can be inhibited. And there are other proteins associated with the viral maturation that can be inhibited. So if you can find any kind of small molecule that can bind to targets associated with each of these processes, these small molecules can then be used as the therapeutics against the SARS-CoV-2 virus. And so in our case, the project that I'll describe is centered around trying to identify small molecules that target a particular virus protease, right? So the idea is if you could knock out this particular, at this stage, then you essentially prevent the virus from completing its life cycle. And so you probably stop the spreading of the virus. So the particular protease that we focus on is it's called the MPRO, which is the main protease. And it's one of two proteases that you can find in the SARS-CoV-2 genome. So the function is it processes a number of viral polypeptides. And so if you're able to stop this or disrupt this processing, right, you can disrupt the replication of the virus. So the good news is that we have structural information for this protease. And so this structure is shown here on the left. And it's relatively high resolution. So we really have a good sense of what the structure of the protease is. And so given structural information, right, one of the things that we can do is to deploy structure-based approaches to try to identify small molecules that can bind to a particular target, in this case, the protease, and then inhibit its function. So in a conventional virtual screening technique workflow, one would assemble or compile a library of small molecules, right? And this could be a library that ranges between tens of thousands to millions of molecules, right? And then you use a computational technique to identify those that are likely to bind to the target. Now, if you were to go through this step, right, and identify such small molecules, there is a lengthy process going from the point at which you identify a molecule that binds to your target, right, and it turning into a drug molecule, right? It's a very long and expensive path to that. So as an alternative, one could attempt a drug repurposing approach, right? And so the idea is that you could start with a library of molecules, right, that are FDA approved and have been advanced through various stages of clinical trials. And then what you do is to screen them against your target, and you prioritize the list and you take the molecules that are at the top of the list that are most likely to interact with your targets. And so this is attractive because then you bypass that lengthy process, because most of these molecules have already gone through extensive testing to look for, to assess toxicity to humans and things along those lines. So this is a really attractive strategy, and a lot of people have actually explored this experimentally, where they take some molecules that are FDA approved or experimental drugs, and they pass them through experimental screens. And the idea then is that once you do this, you can then do some kind of selection, maybe the best molecule, however you define the best molecule, and then advances to the clinic. And so this is a much more efficient approach. If you're going to rely on experimental screening, there's still some cost associated with this process. So another alternative would be to do this virtually, right? Where you use some kind of fast and efficient and cheap computational technique to prioritize the same library, right? And then use that reprioritized library to then do a selection of the best molecules and then advance that to the clinic. And so this latter approach is kind of what we try to execute here. So for structure-based screening, right, we start with a target. And so we know the structure of the target. And so we first identify a region in that target where we want to actually identify a small molecule that it would bind to. So hopefully that we could find a small molecule that binds into this region here. But again, the key is that you have to have structure in it. For the main protease of SARS-CoV-2, we have structure, so we can actually utilize these kind of structure-based approaches. So once you have the structure, right, you can then do some docking. And so you can dock into a cavity. And so every molecule you dock into the cavity, you can determine some binding energy. And its binding energy can then be used as a guide to help you identify small molecules that are likely to bind to your target. Now, this is a computational binding energy. It's something that you could predict, right? So there are some errors involved. But typically, these docking calculations are fast, which means that you could screen a large library of compounds pretty quickly. So some of the challenges associated with this kind of structure-based screening approach, as you probably would anticipate, is it must be accurate, right? And it also must be relatively fast. Now, depending on the stage in which you're applying this, the weighting that you give to these two considerations might be different, right? If you're screening a very, very large library, let's say a library that contains millions of molecules, maybe your first thing that you want to focus on is making sure it's fast. In the intermediate regime, for example, in the regime that we're going to be working with, you want to probably make sure it's accurate because you have less molecules, so speed is less of an issue. It's still important, but it's less of an issue. But in that case, you actually now have to consider both accuracy and speed. Right? So simple scoring functions that are typically used for docking, right? They can take less than a second or seconds to execute, right? But if you do really full quantum mechanical calculations that give you a really physically rigorous way to describe the interactions between a molecule and a target, these calculations could take on the order of several weeks, right? So on the order of a month or so. And so for the kind of project we're involved in, this would not be feasible, right? Doing these full quantum mechanical calculations. So what's the solution? So the solution is a method that has been out in the literature and has been applied to drug discovery in the past, and it's called the Fragment molecular orbital method. And so it's a divide and conquer strategy. And I'm not going to go into a lot of the details associated with this method, but I'm just going to give you a feel for what it does and how it works. So in the center here, what I'm showing you is the ligand. So the ligand is shown in green, and then there are these residues in a pocket that this ligand binds. And these ligands are shown in spheres. So the purple, blue, and red spheres correspond to nearby residues. And what we really want to do is to take this structural information that we get here and estimate some binding energy. So when this binding energy is a number, when it's negative, the more negative the binding energy, the more favorable the interaction, right? Now, if you use a quantum mechanical approach, as I said, to describe or get access to the binding energy would not be feasible in this particular project. And so the Fragment molecular orbital actually breaks up this problem to sub-problems. And so what you do is you treat this problem of determining the binding energy, which is referred to as delta E here, as a pairwise problem where you can express this binding energy as a sum over pairwise interactions between your ligand and each of the residues in your pocket. So in the terminology of FMO, you can treat the ligand as a fragment and each of the residues as additional fragments. And so what you then do is look at, for example, here, the interaction between the ligand and fragment 1, which is the residue 1. And then you can do the same thing here between this residue and residue 8, right? This residue here and residue 7. The ligand and residue 6, and so on and so forth. So it kind of allows you to split up the problem. And the reason why it helps is that you could then treat each of these sub-problems independently of each other so that they can actually be run concurrently. And so it really speeds up the calculation. So from these pairwise interactions, you can then get a total binding energy. And you can also get access to information about the decomposition of the interactions, whether or not it's electrostatically driven or whether there's some electronic effect related to dispersion and so on and so forth. But we're not going to focus on that in this talk today. So again, the Fragment molecular orbital technique is really a divide and conquer strategy that allows you to speed up the calculation, right? Yet still do a quantum mechanical calculation that describes at a high level the interactions between your small molecule and your target. And so here in this project, we've applied it to try to do drug repurposing. So the idea is to take a set of FDA-approved drugs, calculate the FMO binding energies, and then reprioritize the library based on those energies. Now, just to give you a feel for the computational challenge, and I'll list in more detail some of the computational challenges at the end. Most of the calculations take around 24 hours, right? At maximum, they take 24 hours. And we had a library that contained about 2,820 compounds. And so this would require about 67,000 computing hours, if you naively just ran them one after the other, right? But it just meant to give you a scale of the challenge, the computational resources that's required. So a little more details about what we did, right? So we, as I mentioned, we executed these FMO calculations, right? The computational resources that we use for this, right? We got from the COVID-19 HPC consortium. Specifically run these calculations, first testing them out on Jetstream and then finally running the production on Stampede 2. And the goal is, as I kind of alluded to, is to compute the binding energies of these molecules. And just to be complete, right, these molecules that we chose came from this Superdrug 2 library. So this is a library of experimental, FDA-approved experimental drugs that has been compiled, and the reference for that library is given here. So the software we use is GAMES, right? And so GAMES is a quantum mechanical quantum chemistry software tool that allows you to run a variety of quantum mechanical calculations on molecules of varying sizes, right? And it has a wide user base and a wide developer base. And so we utilize it. So in terms of the calculation parameters, right, the theory level of theory we did was at the MP2 level. We used the 631 T-star basis set. We accounted for solvation effects implicitly using the PCM implicit solvent model. And the analysis we did is just simply, once the calculations are complete, we calculate the binding energies and sort our library based on their overall binding energies. And the goal was to identify the small molecules that are predicted to bind to the MPRO with the most favorable interaction, so they have the most negative binding energies. So I'm just going to give you a quick sense of the results. I'm not going to go too deep into the results, but just give you a feel for how we utilize the results here. The first thing we looked at was just the distribution of binding energies. So that's what you're seeing here on this slide. We're looking for molecules in the green zone where you have you have negative binding energies, and you can see most of the molecules actually have favorable binding energies, right? It's just a handful of the molecules actually have binding energies that are favorable, and so this is actually a good thing, right? It means that the calculations are actually selective, right? They're actually saying that there's only a handful of molecules that are predicted to bind to your target. Very often when you use simple scoring functions, many of the molecules are predicted to bind to your target, right? Now, so what we want to do then is to go into this green zone and look at the specific molecules that have the most are predicted to bind tightly to our target, starting at the tail of this distribution. And what I'm going to just show you now is the first 3 hits that we identified. So this is the image of the binding pocket, and these are 2 catalytic residues that are key to the functioning of this protease. I'm just showing you here for reference. And these are some of the sites in the binding pocket that we expect small molecules to bind to. So there is a site S1, S1 prime, S2, and S4. So the first molecule that we found is shown here. This is molecule 1, niclofan. And so it has a binding energy of about negative 11.2 kilocals per mole, right? And it binds in S1, in the S1 region here. And then I'll talk a little bit more about why this molecule is interesting. Next one is trinitrophenol, which binds in the region near S2. And then we have cysteine, which binds in S4, and shown here, right? So these are all the molecules that we expect to bind tightly to this MPRO. Now, if you put them all together, right? If you show them all together, right? This is the region that they occupy. Again, they kind of occupy collectively S1, S2, and S4. And I guess S1 prime is a bit vacant here. So just something a little more about this FMO hit one, right? So when we simply just sorted the library and identified this first molecule, right? And then after the fact, we actually did a literature search and we identified this nicotinamide, right, which is a broad spectrum antiviral that we discovered after the fact is known to actually inhibit replication of SARS, MERS, and Zika virus, right? And so if you compare these two, right, they share some similarities. They have these kinds of dinitro, well, it's actually a chloro phenol, right? And so you have this chlorine here and this nitro group. They're not identical, but they share some similarity, right? So indeed, this molecule is an analog of this molecule here. And so it seems as if we were actually able to identify a molecule in the the superdrug 2 database that resembles a molecule that it turns out actually has some activity against SARS, and not just SARS, but SARS-CoV-1 and SARS-CoV-2 and a bunch of other viruses. So one of the things we might want to do moving forward, right, is first we can search for analogs of our first hit, right? And so this will be an effort to identify new compounds that can have activity against the MPRO. Another thing we can do is also try merging our hits, right? So I showed you this slide where we kind of showed all three hits together, right? One could imagine coming up with a novel molecule that combines each of these three compounds, right? So we can attach them such that they will be able to occupy these three sites. And we could also explore our top hits and identify an additional molecule that occupies this site here. That's something we'll actually look into. If we're working with synthetic chemists, we can then synthesize the molecules and actually test them. Now we could also apply the same kind of approach to other SARS-CoV-2 targets, right? So we focus on MPRO, but there are a number of other targets that one can actually, for which we have structured information that we can actually apply this same kind of technique where we take the crystal structures, dock this superdrug 2 library against those, and then be able to filter the library and identify molecules that might bind to those targets. Now, something that's more long-term is centered around learning from the FMO data. So one of the things that I didn't really, probably isn't obvious from my discussion is the fact that the output from the FMO calculation is very rich, right? So in the current project, we were just using the binding energies to sort the library, right? So this is just this one number associated with each of the compounds. But the FMO calculations gives you access to these pairwise interactions between your in this case, your drug and all of the residues in the pocket. It also gives you access to information about the breakdown of the energetics, the electrostatic component, the dispersion component, the charge transfer component. In addition, it gives you access to information about the atomic charges on the atoms and the ligand and both the receptors. So there's a lot of data that we have access to from these FMO calculations that we can actually use to learn things from. And also we can imagine building predictive models that allow us to reproduce the result of the FMO calculations directly from the 3D structure. And that means that we might actually be able to bypass some of the FMO calculations. And this will be useful if you want to screen a much larger library, a library of the order of millions of molecules. So these are the directions that we're going to take this work in. Now, just to kind of set the stage for Dimuthu's talk, right? Some of the computing challenges we faced here are centered around the scale, right? So we had a 2,820 molecules in our database, and we actually had to carry out twice as many independent calculations. And so all these calculations, as I said, was carried out with gains, right? Half of these correspond to these RNA ligand complexes, right? And required 48 processing cores. And then we also needed job monitoring, right? Because we've experienced some convergence failures in the past when we were testing it out on our own machine locally. And so one needs a strategy for monitoring jobs, checking for conversion failures, and if needed, to resubmit those jobs. Because we run the calculations to get, access to these energies, we have to parse the output. So we needed to kind of automate the parsing process, and so we had to get access to the energetics and the atomic charge data from the log files. So these are the kinds of things that we had to consider or get assistance with. And so I'll stop at this stage and take some questions, and then Dimuthu is gonna take it from there and kind of talk to you about how we kind of address each of these challenges.
Thank you very much, Aaron. That was fantastic, very fascinating. So do we have any questions for our speakers? Yeah, we have one here from Mona. Go ahead.
Oh, okay.
Yeah, just to go ahead and ask.
Hi, thank you very much. That was really interesting. I have a question sort of about the science. I'm curious about how successful this drug repurposing technique has been in the past, and was wondering if you know of examples of its successes.
So I don't have offhand, I don't have a number of examples for you, but I know even at the University of Michigan, we have a drug repurposing center, and they executed a drug repurposing campaign on SARS-CoV-2, and they were able to identify some candidates that showed really good in-cell activity, right? Whether or not those were in turn advanced all the way to the clinic, I don't have a good sense for that. For us, we hope that ultimately that they will be able to be advanced to the clinic, but if not, we might still learn some important information about the kinds of molecules that these targets would bind to, right? And that could probably inspire the design of new molecules that are more potent than these initial hits.
Okay, thank you.
So just a comment, Mona here. I mean, there are published, reports about these repurposed, drugs for various other indications, than what they were originally intended for. And I mean, yeah, this is well known, and they have identified, there is definitely literature available that, I can send you some, because I used in one of my previous presentations at some point, that actually talk about, classes of these, drug, pre-approved drugs for other indications. And that reduces the cost on time of, drug design and drug discovery.
And Madhu, I see you just entered a question in the chat. Do you want to go ahead and ask your question?
Yes, this is Madhu. Is any work going on towards co-crystallizing the protease with the compound you found?
Yeah, so this is work that we're planning to initiate. The truth is that, preparing for this talk, I went into the data and I saw these compounds that I showed you here, and so the next step is to work with collaborators to try to get structural information.
Thank you.
And I actually have a few questions, but I want to make sure that Dimuthu has enough time for his presentation. So let me ask you, do we have time for another question or two?
I'm willing to answer questions, Suresh.
I think, yeah, Bob, please, please go on. I think, yeah, we can adjust.
Okay. Oh, yes. So I just have a few questions. Really fascinating. So I wasn't familiar with the FMO method. This is outside of my area of expertise. But how accurate is it? How does it compare to doing, say, a quantum calculation of the full ligand interacting with the pocket?
Yeah, so a number of studies have kind of shown that they're quite accurate. I don't have the exact number for the accuracy, but I think they're typically within 1 kilocalorie per mole, right? I guess one of the challenges in that kind of work is that the calculations on the full complex is so expensive, right? It's really, really hard to get access to that data, right? Because it takes a very, very long time to actually execute those calculations in the full context of the receptor. I think they're typically within about 1 kilocalorie per mole.
Okay, well that's great. And then another question, with this FMO method, you're taking the problem, you're essentially decomposing into a bunch of pairwise interactions, and then reassembling that to get total energy. And you might have touched on this sort of in your summary, but can you take some of those pairwise interactions and then reuse them? So, for example, if there's a functional group on one of your drugs that also appears on that's also part of another drug. Can you say, hey, we've done that calculation already, or do you need to really do everything from scratch each time?
Right. So yeah. So this is what I touched on about learning from the data, right? So if we have a way of encoding the features that describe the interaction between a given functional group and a nearby residue, right? And then we can say, okay, I've seen this before, right? Then I could go into or essentially train models that could take those features that describe that interaction and map it to a set of FMO energies, right? And that would really speed up the calculation. So that's something we're definitely interested in exploring, because that would, allow us to get access to FMO-like energies without doing the same kinds of calculations.
Right. I mean, you could see, that this being, as you accumulate more and more of these FMO energies to, this really becoming a deep learning problem.
Yep. And that's kind of where we're headed. So there is actually a group. So I think the PIs that kind of really pioneered the use of FMO calculations, they've assembled a database. I think it's called the FMO MD or DB. And so with the goal of publishing or making available FMO data. So we have a similar kind of goal, right, which is to populate a database. And one could imagine leveraging both databases, the one that we would populate and what they already have, to be able to kind of execute this kind of learning you alluded to.
Okay, great. Thank you. So do we have any other questions for Aaron? Right. Otherwise, again, thank you very much. Why don't you go ahead and stop sharing so that we can get it on to Dimuthu's presentation. So go ahead and take it away.
Thanks, Aaron, and thanks for letting us to present here. And thanks everyone for joining the presentation. So can you see my presentation?
Yeah.
Yeah. Okay. So Aaron basically described about the domain problem for the finding a drug for the COVID virus, and in this part of the presentation basically talks about the computation challenge and the the path that we have taken to address the requirements of the FMO calculations that imposed by Aaron and the team. And as the overview of the entire presentation after this is basically we are talking about the challenge that we have seen in terms of the computation, and then what are the resources that we have used for the testing and production calculations, and then how we use IRA, the Science Gateway, to solve this one, and some extensions we have added to support this particular problem, and some results and some future outlook that we are envisioning for other gateways. So this is like a very top overview of the entire project or the problem that we are addressing. And in terms of the science motivation, so the top part, the top row basically talks about the domain problem that Aaron is working on. He has some drug repurposing library, and he needs to calculate all the binding energy across the drug and the protein, probably of the virus, and then filter out what are the possible drugs and then use it in their practical environment. And the scientific computational part comes in between the this drug and the binding energy calculation. So basically, he has like a repository of like 20 drugs, and that needs to be like that needs to be passed to FMO calculation models. Basically, we are using games application to calculate those binding energy and give those values to Aaron to move forward with the research. So that's basically the red dotted part is the one we are addressing in this presentation. In terms of the nature of the computation, I think Aaron briefly touched.
Cheaper is good for me. You think it's okay? Yeah.
I'll go ahead and do them. Somebody accidentally unmuted, and I'll go ahead and mute their mic.
All right. Okay. So, so in terms of the computational nature, so we have like basically two types of computation. First type is the calculating the FMO energy of the drugs. We call it like ligands. So there are like 20 drugs that I have selected, and to calculate the FMO energy for each and every drug, for one drug, it takes average, like the initial testing showed that it takes like around like 5 minutes on a 48 core machine to calculate. So basically like it's like a magnitude of 20, like if you are running it serial manner. And then we use that same drug and calculate an interaction between the protein that we are used to measure the docking, and we call like complexes. And to calculate the FMO energy on that particular substance, it takes like 7 hours, and which is understandable considering the complexity of the entire substance on a 48 core machine. And however, like these are like two main types of the computations we have to do, and it has to be like the multiplied by the number of ligands or the complexes that we need to run. And the solution that we came up with is the static parameter sweeping, because we have like the configurations of the computations are most of the time same, but the parameters or the inputs are the only one who's going to be like a different ones. So we can replicate the same problem on different nodes or the different machines on the cluster and run them in parallelly. So if you look at the right image, it's basically like shows the abstract view of how we are going to scale the computation. So basically, we are in the cluster, we are submitting like one parent job which contains all the information of the drug molecules or the complexes that we need to compute, and those are getting scaled across the available nodes of the cluster to calculate each and every FMO energy of the molecule. So the rest of the next two slides, I'm going to talk about the computational infrastructure we have used. So initially we used JStream virtual clusters to set up the environment and test the games applications and get the right output, test whether we are getting the right outputs. So basically we use JStream as a test bed and the debugging infrastructure to initially set up the whole platform. And understandably, we had like very limited hardware resources, and we calculated like just for our reference how much time it will go, like if we just use JStream. So basically, to at least calculate the complexes, it takes like 828 days theoretically, which is not like the right approach to do. So basically, JStream is only the test bed for us to check the entire platform. So once we have like set up entire like the platform, then we move to Stampede 2, which contains highly performing nodes. And luckily, we got the access to SKX normal queue, which contains Skylake Intel nodes, which contain like 48 cores at a node, and we got access to like 120 nodes. And we're really thankful to Kent Milfeld from Tech Team, and I think he's on the call as well. And I'm just getting this chance to thank him to providing us this facility, and he worked with us to get the resources and making like custom modification to scale the jobs on the Stampede 2 side. And in terms of the scaling, we use some custom node binding mechanism on Stampede 2, because on Stampede 2 we don't have the access to SERM job arrays to do it natively scale at the SERM level. So we are using SERM, and inside that we are using a custom mechanism to scale the jobs, and I will talk about it later in the presentation. And theoretically, like we can run 120 jobs at a time, given the number of nodes we have. And in each node, we are running a one calculation at a time. So if we calculate, if we get the theoretical number of the runtime for the protein ligand complexes, it takes around like 168 hours, and considering we are running 120 in parallel. And it takes like 7 days to calculate entire complexes. And for the ligands, the estimation was like 2 hours to calculate the entire batch. And obviously these two, obviously in the practical it took more time, but we need to have like a baseline to understand about the scale of the problem. So then we use Science Gateway to orchestrate all of these, because as you understand, it's a like highly parallel computational workload, and amount of the parallelism we are working on, it's very complicated to submit jobs, do the load balancing, and even like fault tolerance, monitoring all these sub jobs, and even like data transfers. Those are really complicated work while we are working on the main science problem. So we let the Science Gateway to take care of that one, and PI, basically Aaron and the team had to work on data preparation and just submit to the gateways, and the gateways do the rest of the work. For this one, we use Apache Irauta Gateway Framework, and SciGap is the software as a service platform of the Apache Irauta. Right now, SciGap has more than like 45 gateways deployed, and more than like 230 applications supported with 70 plus computation resources. And right now we have like more than 4,700 users registered on the SciGap platform using all the computation resources that we provide. And if you look at like the Apache Irauta platform as a very abstract view, you can connect many computational and storage resources of your own or some cloud or national-based resources to the platform. In addition to that, it provides the ability to connect all the instruments. For example, if it is like MRI data management or some image analysis tools, you can just connect it to the platform. And from the other end, you have the portals to access all of them. And furthermore, we are supporting to deploy those portals in our infrastructure so that you don't have to worry about the availability of the portals on the 24 hours. So these are like some of the collaborations we are working on. In the interest of time, I'm just trying to skip of this one. Basically, it's just like we are basically providing a lot of we have supported a lot of collaborations in different disciplines, and you can go to this URL and look at the other ones we are going to support. And coming back to the problem, so we use Apache Irauta, but still we couldn't use it as it is, because this problem is like a different scalable problem. Basically in Irauta, we have like a concept called one experiment, one job. Like user submitting one experiment and it's going to submit it as a one job. But here, the problem is one experiment and it could be scaled into number of jobs. So in that case, we have to support this parameter sweeping aspect in the job orchestrator level of Irauta. And because we were working on all the SERM-based schedulers for this particular one, two approaches we had was to use job arrays, and we used job arrays in JStream. And in the Stampede 2, we use like a custom node binding mechanism based on MPI. And then the other challenge was to how to prepare the workspaces for these jobs on the cluster side. Basically, like how to create the folder structure for the jobs and how to place the inputs over there and how to get the outputs from there and where to write the standard outs of the jobs, and all the preparing workspaces was also another extension we had to work on. And the tricky challenge after that was how to monitor those subjobs, because the scheduler itself doesn't support to monitor subjobs other than you are just go to the scheduler and follow the status. So we had to come up with a way to get the event-based notification for these subjobs we spin up in the cluster. So at the end of the execution, we needed to pass the outputs, which are like the useful for the downstream analysis for Aaron and the team. So we are doing additional data passing workflows in our infrastructure to pass the outputs generated from the cluster. So this is basically, this slide basically covers how we create the environment. So users are submitting an experiment which contains all the input files, and the gateway connects to the compute resource and create the folder structure. Like if there are 4 jobs, create 4 directories in the main working directory and place all the input files in the right order, and let the scheduler to submit the jobs after that. So this one basically covers how we did the parameter sweeping on Jetstream. We use Slurm job arrays to do that one. We create a one parent job description in the Slurm script. Then the Slurm script, we specify how many jobs you need to run in the array section. And then if you can remember, like in the previous slide, we are creating the directory structures for each and every job. So here we are specifying, okay, each subjob created from the Slurm job array gets an ID. That the %A means the job ID you are getting, and you are and it's using this working directory to write the standard error and standard out of each subjob. Then we go to the right folder and then execute the games application inside that particular folder. And in the Stampede 2 side, the static parameter sweeping was a little challenging because we didn't have the Slurm job array supported natively. So the other one was to use a launcher or some other custom mechanism. In this case, Kent helped us to come up with a specific, like the script, which works for this SKX normal queue. So basically what it does is it gets all the node list available for the SKX normal queue and iterate through all the jobs that we need to run and bind those jobs into the right node. So this is a very optimized way for this particular use case. So because we exactly know that each job is scheduled at the right node, and each node getting only one job to run at a time. So this is the approach we followed for the Stampede 2. However, like the both approaches, computation model was similar. We are submitting a one job, and they also are getting scale at the cluster level. But the challenge then was how to monitor those subjobs, because the scheduler doesn't give the notification for the completion for those subjobs. It just gives the notification from the main job completes. To address that one, we had to come up with a way to to get the notification. So the approach was to programmatically inject curl commands to the before top and the bottom of the script, so that when the job is getting executed, those curl commands are also getting executed. And if you look at the top curl command, it has the annotation of the job status and the parent job ID, child job ID. And the end of the curl command also, we are getting the completed notification and some same parameters we see in the top one. And those curl commands are directed to a HTTP server we deploy in our infrastructure. And once the server gets the curl invocation, it sends it to the Airavata job orchestrator. The job orchestrator knows how to map those curl commands into the right subjob. So if it is a completion notification, it starts to fetch the output from the cluster for that particular subjob. So the advantage of this approach is that we don't wait until the entire batch to be completed. We are running on demand, like once each and every subjob completes, we are getting the output so that the end users can see the outputs before the entire job batch completes. But in this approach also, like there's an issue because we are relying on curl and it's basically on TCP. And if the network is unstable, TCP might not work perfectly and you might lose the job notifications. However, like we need a way to make the entire system eventually consistent. It means like if there are some notification, if some notification were lost, however, like at the end of the day, it should be able to recover. So if in this diagram, child tree notification was lost on the network, so the workflows, the post workflows means the data output data staging from cluster to the gateway work only for the child 1, 4, and 2. So after some time, once all the jobs are completed, parent job sends an email to our email account. So then we know, okay, the parent job was completed, but still the child tree was missing. So then the job orchestrator talks to the cluster and see whether the child tree was completed or not. If it was completed, then it wraps up the missing job notification and start to get the output after that. So using this approach, we are getting fast outputs and at the end of the eventual consistency for the missing job notification. Then once we get the outputs, Aaron wanted to convert those outputs to domain-specific format for them to do downstream processing. So they gave us some conversion scripts. We had a framework called data passing framework. So we are injecting those scripts as wrapped Docker containers, and the data passing framework loads those output files generated from the cluster and convert them using the scripts that Aaron provides. And then we get the new outputs or the some user-friendly outputs, and we are putting into the gateway storage. And at the end of the day, this PI is getting raw outputs generated from the cluster and all the pass outputs generated from our infrastructure. So he can use either of them. So these are some of the observations. The Stampede2 side, for the ligands, we submit like total 24 jobs across 29 completed experiments. And some 7 experiments failed due to some queue size issues, some SSH errors. And if you look at these graph here, the time for the computation on the ligand, it like somewhere like lies around like 0 to 15 minutes. So most of the frequency you can see hit around like 5 minutes. So our initial estimation was correct in a way. And for the complex calculation, we submit like 89 jobs and across 28 completed experiments. And some experiment failed due to some SSH and allocation. So we had to resubmit those. So the computation time for the complex, it little like distributed compared to the ligands. I start with like maybe like 6 hours to like 14 hours. Some jobs took 14 hours, and most of them were like focused around like 8 to 9 hours. And on the jet stream, we didn't do further analysis because it's only for the testing purposes. Altogether, like we have like 30 completed experiments and 19 failed experiments, which is expected as we were working on setting up the environment and testing the framework. And these are some of the challenges we have observed. First, we had like we were working on different games version on different machines. For example, on Stampede 2, we had one version, JStream, we had one version, and Indra's machine, there was one version. And interestingly, like these different versions were giving different FMO calculation for the same input. So we had to compile the same version on all the machines to get the consistent output. And the other challenge we have observed was not having some standard, like the common mechanism to scale jobs on compute resources. So we had to come up with some custom mechanism to scale on the production cluster we were working on. Looking at the future, so first thing we need to work on, like to provide some user-friendly ways to integrate the data parsers. Right now, it's more developer-centered one. So the developer has to integrate into the framework. But in future, we are planning to provide more interfaces for the PI or the user to directly integrate their parsers into the framework. And in addition to that, we plan to provide some more flexibility of the computational resource types, possibly like the scheduler integrations. And most importantly, learning from this particular project, we plan to generalize all these data parallel parameter sweep code that we have implemented for this particular one to other Airavata gateways, so that other users also can use the parameter sweeping approach for their experiments. And the other one is we need to think about like how to do more efficient resource utilization, especially like in the case of Stampede 2, we can, like if you look at the distribution here, like there are some jobs running 14 hours, some jobs running like 5 hours. Like if one job runs 14 hours and the rest of the nodes are idle, we can utilize the rest of the nodes for the next batch. So those things we need to further experiment and research. And we are planning to do it in future in a generic way. I think that's the end of the presentation. Maybe like we can wait for the question section.
Great, thank you. Thank you very much, Dimuthu. We are a little bit over, but do we have any questions? So I have one. So you use a mix of Jetstream and then Stampede 2, and of course soon IU is going to be deploying Jetstream 2, which, if I remember right, it increases their cloud resources by roughly a factor of 16. Would you consider moving the entire workload just back to Jetstream 2 once that's deployed?
So for this one, Bob, I think we are done with all the calculations.
Okay.
So the framework as a whole, we don't like bind to a particular resource. We are like providing like more ways to integrate with the resources. So if the Jetstream 2 support job arrays, we can do the same way we have done with the Jetstream.
Okay. Any other questions? I was going to say for any of our speakers, but I see that Aaron had to jump off, but he left his email address if you wanted to follow up. All right. Well, if not, again, thank you. Thank you so much to all of our speakers. This was fascinating today. The ECSS Symposium is professionally often my favorite hour of the month. Just love seeing what you're all doing. So with that, unless there are any more questions, any more comments, let's just thank our speakers again and hope to see you again in June.